Hello, and welcome to this virtual Bioline Academy talk about biocontrol advice. We are happy to be teamed up with Carlin Horticultural Supplies ProGreen Plus, and we're excited to bring you some considerations when putting together a biocontrol program. My name is Daryl Johnson, and I am Bioline's Regional Manager for the Eastern United States. I have a degree in ornamental horticulture from Ferris State University in Michigan and over 15 years of experience working in various facets of horticulture. Before we get into the rest of the presentation, I would like to take a few moments to introduce Bioline AgriSciences. Bioline AgriSciences is part of a large French cooperative group called the In Vivo Group, which represents over 50% of the growers in France. We have been working in the biocontrol industry for over 44 years now. And during that time, we have been able to put together 8 biofactories around the world, 4 of which are in California. We understand that improving our products is very important to our growth and the success of our grower partners. We reinvest over 10% of our earnings back into our products that make our bio solutions superior and more effective for growers. Here we see a map of all the locations of our biofactories. Our biofactories are kept very clean. They're run as laboratories to ensure that we're producing the highest quality beneficial insects and mites for our growers. As we go through all the considerations to keep in mind when developing a biocontrol program, I also think it's important that we review the reasons why we might be using biocontrols. By no means is this a comprehensive list, but it covers some of the most important aspects of why one might use biocontrols. Biocontrols are easy to use, and they're definitely a viable alternative to conventional controls, especially because conventional controls are losing efficacy and there are more and more restrictions on them. There is no re-entry interval when using biocontrols from Bioline. They can be applied at any time when there are workers and or customers nearby. There are no personal protective equipment requirements for release of our products. There are no risks of pollution, chemical exposure, or phytotoxicity, and it is a marketable attribute for retail growers. Things to consider when we are developing a biocontrol program. First off, we need to think prevention. We want to be ahead of these pests before they get a foothold within our crops. Secondly, we want to have a plan and we want it to be adaptable. Additional considerations: the pests that are going to be in our crops, what control measures are available for those pests, what types of release methods and frequencies, the crop type and location, the crop length, and also the time of year. Next, we will review the 4 most common pests that we encounter in crops, whether those crops be ornamentals, food crops, or medicinal crops. The first pest we will review are thrips. This is a list of the 5 most common thrips that we see impacting ornamental crops. For this presentation, we will be focusing on western flower thrips. I like this slide because it showcases how quickly thrips can reproduce within a crop. Over the course of 60 days, at a temperature of 68 degrees Fahrenheit, 1 thrips can turn into almost 6,000 thrips. That's why it's important that we get controls into the crop as soon as possible. We see success when we have biological controls before we have pests present within the crop. If we wait 30, 60 days down the line, we will definitely be disappointed. It is important to understand the life cycle of thrips to understand how we are going to attack this pest within the crop using biocontrols. First, it starts off with an egg within the leaf tissue itself. We do not have any biocontrols available to attack this life stage. After it hatches, it will produce 2 larval stages. These larval stages feed on plant tissue that is above the soil line. After they are completing their larval stages on the plant tissue, they will drop off of the plant and pupate in the soil. There is a pre-pupa and a pupa stage in the soil or any growing medium. When they're finished pupating in the soil or growing media, they will emerge as a reproductive flying adult. There are many different controls that we utilize to combat thrips within a crop. We utilize insects, mites, nematodes, both above and below the soil line. We also use physical trapping to remove adults from the crop. A good thing about thrips is that we have solutions for all life stages of thrips. The lines in this slide represent the soil line of the crop. Above the blue line, we see Oraline, Amblyline, and Starsky. These are all predators that we use on the plant tissue that is above the soil line. Exhibit line SF, Hypoline, Staphyline, all represent predators and solutions that we use below the soil line. Two predatory mites that we use above the soil line are Amblysaeus cucumerus and Swerskii, or our trade names Amblyline and Starsky. Cucumerus we use when conditions are cool. I like to remember it with the phrase cool as a cucumber, or in this case, cool as a cucumerus. Starsky starts with an S. When somebody touches something hot and it sizzles, sizzle starts with an S, so does Swerskii or Starsky. We use Starsky when conditions are hot. Cold conditions can be considered when 24-hour average temperatures are below 74 degrees Fahrenheit, and warm conditions or hot conditions being above 74 degrees Fahrenheit, 24-hour average. Both of these mites will attack the first larval stage of thrips, and Starsky will also attack the second larval stage of thrips. If whiteflies are a concern in your crop, Starsky is your first line of defense with whitefly control. If broad mites are a concern, Cucumerus will actually go after broad mites as well. These are great examples of what Amblysaeus cucumerus and or Swerskii can look like in the crop. They have a seeking and ambush behavior within the crop. When scouting, you can find them in flowers or where veins meet larger veins underneath the leaf surface. Delocia coriaria, or our product name Staphyline, is an excellent predator to include in any biocontrol program. It is a soil-dwelling predator that not only eats the pre-pupa and pupa stages of western flower thrips, but will also eat the larval stages of fungus gnats and shore flies. It's ideal for use in propagation, but can be used in all stages of plant production. Staphyline can be used starting at 54 degrees Fahrenheit. Hypoline, or Stratiolaelipsimitus, is an excellent predatory mite that we utilize below the soil line. It is effective in controlling thrips pupa and prepupa stages. It also eats fungus gnat larvae. We can start using this predator right at propagation or right at transplant. Not only does it go after those pests that I just mentioned, but it also will attack root aphid, which we see in this video. We can start using Bioline at 45 degrees Fahrenheit. Bioline, or Aureus insidiosus, is a predatory insect that eats larvae and adults of thrips. In fact, it is the only predator that we offer that will eat adult thrips. We can start using this predator when temperatures are in the mid-50s. It also feeds on other small pests such as spider mites and aphids. In these photos, we can see examples of the different life stages of Aureus within the crop. From its youngest to its oldest stage of development, Aureus goes through many different color ranges and shapes. Nematodes are another useful tool when combating thrips within your crop. Exhibit line SF, or Steinernema feltiae, is the variety that we use for western flower thrips and for fungus gnats. These little guys can be used as a foliar spray or as a soil drench. The optimum temperature range is 54 to 83 degrees Fahrenheit. Once we start getting outside of that range, conditions can be harmful for the nematodes. We also offer other varieties of nematodes when we're looking to control different pests such as shore flies, vine weevils, and cutworms, to name a few. This is what we can expect to see when we look at thrips or nematodes under magnification. When we're looking at the nematodes under magnification and we see them in a squiggly or S pattern, we know that the nematodes are active and alive. If the nematodes are pinned straight, then we know that those nematodes are dead. Trapline T+ is an innovative tool that we implement when combating thrips within the crop. Trapline T+ is sticky tape that is utilized for mass trapping of pests within a crop. It utilizes a unique color and visual pattern that is combined with an adhesive that is impregnated with a thrips sex aggregation pheromone that is highly effective at drawing thrips out of the crop. Each thrip adult that we are trapping on the tape is one less adult that is reproducing within your crop. Soil pests, like fungus gnats and shore flies, can feed on your root tissue below the soil line. Not only is this detrimental to the development of the plant itself, but it opens up entry points for pathogens like Pythium or Phytophthora to enter your crop. When combating fungus gnats within the soil, we utilize some of the same predators that we talked about when controlling thrips within the soil. Bioline, Staphyline, and Exhibit line are all very useful and effective when controlling soil pests like fungus gnats and shore flies. Next in the list of horrible pests to find within your crop is aphids. Aphids are some of the most well-known pests, both with growers and home gardeners. The reason for this is because of their size. They're one of the larger pests that we're talking about today and can be easily seen in flowering crops. This leads to growers having a very low tolerance for this pest within the crop, which means we need to be even more proactive in our control measures. An interesting fact about aphids is that they do not need a male to reproduce. They reproduce asexually. Each aphid you see is female and born with live young already inside them. This makes it extremely important to identify populations quickly or have a robust prevention program in place. Things can get out of hand very quickly, especially when temperatures are very hot. I like to call aphids the mosquitoes of the plant world. They have a needle-like mouthpart that penetrates the leaf tissue. After penetrating the leaf tissue, they extract nutrients from the phloem, which can impact the growth of the plant negatively, sometimes distorting the growth itself. Here we can see some examples of what aphids can do when left unattended. Their populations grow so large that their excrement can actually grow a fungal organism called sooty mold. These happen to be examples on sweet peppers grown in a greenhouse, but this can also be found in ornamentals too, on populations that go undetected. I mentioned just a couple slides ago that aphids can cause distortion in leaf tissue or plant growth. Here are some examples showcasing that fact. Things can get ugly very quickly, especially in the growing tips of the plants. Although aphids tend to be some of the larger pests we see within the crop, they can easily go undetected. They are often green, but color can vary within the colony depending on the type of food that they're eating or the type of aphid that we're looking at. Some of the most common aphids we see within the greenhouse are green peach aphid, melon cotton aphid, potato aphid, and foxglove aphid. As I mentioned before, they reproduce asexually, giving birth to live nymphs, and they cause a lot of damage to the crop. We talked about honeydew, destruction of plants. Two items that we did not talk about are molted skins, which are sometimes seen before aphids are detected. And aphids can also transmit virus within the crop. When using preventative measures of biocontrol for aphids within the crop, we talk about aphids in two different categories: large species aphid and small species aphid. Two of the most common small species aphid are green peach aphid and cotton melon aphid. Two of the most common large species aphids are foxglove aphid and potato rose aphid. When controlling aphids, there are a few different options we have at our disposal. Parasitoids on the left-hand side of this diagram and predators on the right-hand side of this diagram. We use parasitoids when controlling aphids in a preventative way. Parasitoids are parasites that will end up killing their host. Aphidius colemanni, Aphidius irvi, and Aphelinus abdominalis are three of the best parasitoids that we have to use. Colemanni will go after small species aphids like green peach aphid and cotton melon aphid. Irvi will go after large species aphid like potato rose aphid and foxglove aphid. When we start using Aphelinus abdominalis, we stop talking about size categories because this parasitoid goes after multiple species of aphids that covers both the large and small species that we are concerned with. This diagram illustrates the life cycle of the parasitoids that we just looked at. It starts with the adult female laying an egg within the aphid. After a few days, the parasitoid will hatch inside of the aphid. When that larval stage starts consuming the aphid, it turns it into a dead parasitized mummy. After the parasitic wasp is done developing, it will chew a circular hole in the back of the aphid mummy and emerge as a reproductive adult. This completes its life cycle. Here we see some real-life examples of that life cycle in action. When growers see or report aphids within the crop, it doesn't necessarily mean that it is a bad aphid. Sometimes aphid mummies are perceived to be live aphids, which in fact they are not, and they're actually helping control the aphid population within the crop itself. Each aphid parasitoid produces a different looking mummy. Aphidius colemanni, or apheline, produces a golden brown mummy. Aphidius urvi, or urviline, produces a golden yellowish mummy. Both of these mummies are spherical or change shape, which is significant. Apheline, or Aphelinus abdominalis, produces a black mummy. All the while, that mummy does not change shape. It looks the very same shape and size as the live aphid did. Aphid predators are different than the parasitoids. These predators will actually consume aphids off of the plants. Predators are typically used for correction type situations, although can be used in a preventative manner as well. Green lacewing, or chrysaline, is an excellent predator of aphids. An important note to make about chrysaline is that its larval stage is the only stage of development that will be consuming aphids. Adeline, or lady beetle, or ladybug, is an excellent predator as well. All of its life stages consume aphids. Aphidoline, last but not least, is also a great predator when it comes to finding populations of aphids in the crop. Its job as an adult midge is to fly around the crop looking for aphid colonies. When it finds an aphid colony, it will then proceed to lay its eggs nearby. When its eggs hatch, the young larval stages will consume the aphids, reducing the population within the crop. All of these predators are indiscriminate when it comes to the types of aphids that they will eat. They will eat all large and small types of aphids. Last and certainly not least when it comes to destruction of crops is two-spotted spider mites. Spider mites are relatively small, being 2 to 3 millimeters in size, with 2 large dark stains on their side. They can vary in color from green to red depending on the crop that they're eating or if they're in a diapause stage. There are 5 stages of development: egg, larva, 2 nymphs, and an adult. The eggs are small, transparent, and round. Two-spotted spider mites will inhabit the whole plant. They will feed and damage all parts of the plant, including flowers. They produce a white stain or yellow feeding spots on the leaf and flower tissue of a crop. Here are some classic examples of two-spotted spider mite damage within a crop. The yellow marking or staining on the leaf can sometimes be mistaken for thrips, so be extra careful when you're scouting your crop after the detection of damage. The other photos exhibit webbing within the crop, which is very severe damage and sometimes irreparable if conditions are left to get to this point. Similarly to thrips, two-spotted spider mites do not need a male to reproduce. In fact, if a female finds herself without a mate within the crop, she will proceed to produce male eggs, after which those males hatch. She will then reproduce with her young, creating a population that grows very quickly. Again, similar to thrips, left without intervention, one mite can turn into a ridiculous 140,000 mites in the course of 48 days, which is simply amazing, at the temperature of 71 degrees Fahrenheit, which is not very warm when we consider most of our crops are being grown within the greenhouse. When it comes to controlling two-spotted spider mites, we have 4 excellent options. Bioline, which is our specialist for two-spotted spider mites. It only feeds on two-spot. Anderline, which is a great preventative control for two-spotted spider mites. It's one of my favorites. It will also feed on thrips larvae, L1, and it also goes after hemp russet mite or any sort of verified mite. Feltaline is another great predator for two-spotted spider mite. It is a midge, and its young will eat two-spotted spider mite. And then last but certainly not least is Californialine, which is another predatory mite, a generalist as well, and we tend to use this predator when conditions are very dry. It will reproduce easily at drier conditions. This slide illustrates the wide range in delivery options that we have when it comes to putting bios to work in your crop. Blister packs, sachets, loose material, capsules, and dispensers are all ways that we can release beneficial insects and mites into your crop. We divide our release systems into 2 different groups, quick release systems and controlled release systems. The quick release systems encompass loose material and blister packs. This method provides immediate dose and an increase in predatory mite populations within the crop. This is best for the initial treatment and/or hot spots of pests in the crop. This can be used with mechanized applications such as a blower, and the recommendation is to make weekly applications if using quick release methods. Controlled release systems can be seen as sachets or bugline. These sachets contain a breeding population of mites that will sustain a population for the course of 3 to 6 weeks. Sachet life is dependent upon the environmental conditions, mainly humidity. Here are examples of our quick release systems, our innovative blister packs, and our loose material in liter tubes or 5-liter bags. Blowers are a common tool to use when releasing loose material within the crop. Here are a couple of examples of creations made by growers to help save them time and effectively release beneficials into the crop. Sachets, capsules, and dispensers are our controlled release systems that can help save labor and keep consistent release of predators within the crop. What is in a sachet, you might ask? It's a great question, and it's a very common question we receive. Inside of our sachets is a brand type material that contains a yeast. That yeast acts as a food for prey mites within the sachet, and those prey mites act as food for the predatory mites that we're looking to release within the crop. This creates a breeding culture of predatory mites that releases over the course of 3 to 6 weeks, depending on your humidity. This slide illustrates the capacity of what one of our sachets can actually produce. Bioline, or cucumeris, starts out with 250 mites, and over the course of 4 to 6 weeks can produce 700 to 1500 mites. Starsky, which starts out with 125 mites, over the course of 4 to 6 weeks will produce 500 to 1,000 mites. We have options for all different scenarios, all different types of sachets to overcome the obstacles of a variety of different crops, whether you're growing ornamentals or food crops, indoor or outdoor, could be medicinal crops, you name it, we have a solution for that type of crop. Bugline is my most favorite release system that we have to offer at Bioline. It is awesome at saving labor because what it does is it takes our sachet technology and puts it into a ribbon format. This ribbon then can be pulled through the crop or laid right on top of a crop, reducing labor because no longer are we placing sachets every so often or hanging them on, say, on the plants. We are utilizing the efficiency of a ribbon and getting the work done quicker. Here we see Bugline being utilized to make crop of dahlia. This particular grower stopped growing dahlia years ago because they could not get enough control of western flower thrifts within their crop. By utilizing biocontrol and Bugline, they were able to turn this crop on again and make it a profitable sale for the year. Blister packs are an innovative release system that helps us meter the release of our predatory mites and parasitoids evenly throughout the crop. We started out using this release system for our parasitoids, for aphids and whiteflies, but also found it extremely useful for our predatory mites. Phytoline, Starsky, Amblyline, mixing both Persimmilus and Californicus in our PerCal mix, Californiline and Anderline are all available in this style of release system. We went through a ton of information during this presentation, and I want to thank you for sticking with me until the end here. I promise you that we're almost finished. Okay, some key points to take away with you today. The first, we'll start off with 2 rules of thumb concerning our release methods or release systems. Quick release systems, which are our loose material and our blister packs, those are recommended to be released weekly. Our controlled release systems, like our sachets and Bugline, those are meant to be released every 3 to 6 weeks, depending on your humidity. Always plan early and commit to the program. Do not wait until you have a problem. Biocontrol is a preventative mindset. We want to make sure you're understanding the pests and the controls available to combat those pests. We want to prepare a release schedule and adjust that schedule as we go. None of this is written in stone, and we need to make sure we're adjusting to fit your facility and growing style. And lastly, please contact your sales rep at Carlin or your IPM technical specialist at Bioline, that very well may be me, for any questions or support that you may need. We are here for your success, and we're here to show you a healthier way to control pests within your crop. Thank you again, and thanks to Carlin for inviting us to speak at their BuyerFest event. We're very happy to be partnered with Carlin, and even happier to help the growers out find a better solution for pest control. It's not always easy, but we're here to help. So please reach out, send an email, or give a call. We'll be happy to help you. Thanks again, and we hope to see you soon. Bye-bye.